Hello aspirants, and a very warm welcome to this high-yield explainer right here on the Rungta IAS Academy YouTube channel. Today, we are absolutely thrilled to decode a groundbreaking new study on recent human evolution. Believe me, this is incredibly relevant for your upcoming UPSC examinations.
Spot on, sir. Welcome, everyone. Clearing this exam isn't just about reading textbooks. It's about seamlessly connecting cutting-edge current research directly to your syllabus. And this recent Harvard Medical School study, it is an absolute goldmine of data on ancient genomes. Let's dive right in and see exactly how it fits into your preparation.
Right off the bat, let's map this out. Notice how perfectly this entire topic aligns with the biotechnology module of your GS Paper 3 syllabus.
Absolutely. When the UPSC asks you about biotechnology applications in the mains exam, you can't just rely on old data. You need solid, contemporary examples. This specific study, which was actually published in the journal Nature, sequenced DNA from human skeletal remains spanning thousands of years. It really is the ultimate case study to quote in your answers.
So let's talk numbers, because the scale here is just staggering. 15,836 versus 6,438. Those figures literally represent the massive ambition of this research. Harvard scientists took over 15,000 ancient DNA sequences from all across Western Eurasia and cross-referenced them against more than 6,000 modern individuals from those exact same regions. To put that into perspective, it's the largest survey of ancient human genomes ever conducted.
So what exactly did sifting through all those thousands of genomes actually reveal? In a nutshell, sustained natural selection.
Exactly right. By running advanced statistical models, the researchers prove beyond a doubt that the specific genetic shifts we've seen over the past 8 to 10 millennia were driven strictly by active, sustained natural selection. This wasn't just random genetic drift, and it wasn't just caused by populations migrating around. It was evolution happening right before our eyes.
Okay, aspirants, I need you to pay close attention right now because we are shifting gears to a highly probable prelims focus. Think about it. How do scientists actually know a random skeleton is exactly 18,000 years old? The answer is carbon dating.
And this is such a brilliant illustration of a core static concept you absolutely must know. Here's how it works. Cosmic rays constantly collide with nitrogen in the atmosphere to create carbon-14. When any plant, animal, or human is alive, we are constantly absorbing it. But the moment we die, that absorption stops, and the level of carbon-14 begins to drop.
Precisely. Through the process of radioactive decay, that carbon-14 slowly turns back into nitrogen. And it does this at a very predictable rate with a specific half-life of 5,730 years. Make sure you keep that exact mechanism crystal clear in your mind for the prelims. Now, for a quick science and tech technical note, how is this tiny amount of carbon physically measured in bones that are, say, 18,000 years old? Scientists use an incredibly precise instrument called a mass spectrometer. This highly sensitive device essentially measures the ratio of radioactive carbon-14 against the stable carbon-12 and 13 isotopes. That ratio is what allows them to estimate the skeleton's precise age. Right, aspirants, I highly recommend you take a screenshot or jot this down immediately. Consider this your high-yield mains cheat sheet. Directly quoting these specific genetic variants in your mains answers is a surefire way to fetch those extra half marks that make all the difference.
For sure, sir. We've laid out the specific ABO, HLA, DQ1, and CCR5 genes, mapping them directly to their evolutionary traits based on the Nature study. Let's unpack what each of these actually means.
First up, let's look at the ABO gene, which dictates our blood types. The data shows that over the last 6,000 years, the B variant became significantly more common among West Eurasians. But at the exact same time, there was a concomitant decrease in the A variant. Why? Well, it seems populations really benefit from maintaining an optimal balance of these blood types. That genetic diversity allows a population to efficiently defend against constantly changing pathogenic exposures. Next, we have what researchers are calling the HLA-DQB1 paradox. This particular variant actually makes people highly susceptible to celiac disease. That's the condition where eating gluten triggers the immune system to attack the small intestine.
Now, logically, you'd think natural selection would weed out a disease variant, right? Yet, inexplicably, over the past 4,000 years, this specific variant spiked dramatically, going from 0% to 20% of the population.
And here's the real kicker. Researchers explicitly clarified that this massive 20% spike was not solely driven by the rise of agriculture 10,000 years ago, as you might assume with a gluten-related issue. The true evolutionary driver behind this paradox still remains a complete mystery. Moving forward in the timeline, around 8,000 years ago, early humans began heavily selecting for gene variants that produce lighter skin tones and pigmented hair. This is a textbook example of environmental adaptation. You see, early farmers moving north had diets quite low in vitamin D. In these new, colder regions with significantly less sunlight, lighter skin became an absolute biological necessity just to efficiently synthesize vitamin D from whatever weak sun exposure they got. Now, this next finding is genuinely an astonishing biological anomaly. If an individual possesses 2 copies of the 32 variant of the CCR5 gene, they are essentially completely resistant to HIV-1 infection. But wait, here's the mind-blowing part. The frequency of this incredible HIV-resisting variant actually spiked thousands and thousands of years ago.
Exactly. It predated the actual origin of the HIV virus by millennia. It's as if nature was actively arming our ancestors with a powerful genetic shield against some totally unknown ancient pathogen long before HIV even entered the picture.
Interestingly, the researchers didn't just look at diseases. They also discovered signals of ancient natural selection in gene combinations that we typically associate with very modern traits today. Yes, we're talking about genetic combinations linked to things like intelligence test performance, total years of schooling, household income, and incredibly, even a faster walking pace.
And get this, genetic variants associated with an affinity for smoking were heavily selected against in those ancient times, which is just incredible because tobacco was completely unknown in Eurasia until less than 600 years ago. That genetic aversion was somehow already hardwired into us.
All right, let's pivot strategically here to your UPSC interview preparation, because if the board asks you to contextualize this study for India, you have to be ready. As the eminent scientist Dr. D.P. Kazbekar points out, modern South Asians possess an incredibly diverse, deeply interwoven genetic matrix.
Absolutely right, sir. Our ancestry isn't just one linear line. It's a wonderfully complex tapestry. It includes deep genetic contributions from Iranian Neolithic farmers, Western steppe herders, indigenous Eastern Eurasian ancestors, which encompasses the ancient ancestral South Indians, as well as significant contributions from East Asian, Southeast Asian, and Australian ancestors. It's the ultimate melting pot.
Which brings us directly to a highly provocative, policy-oriented question, perfect for discussing with your interview board. If the West has successfully mapped 15,000 ancient genomes to understand their past, what exactly is preventing India from officially assembling our own ancient genetic legacy? A comprehensive indigenous ancient DNA study here in India would be an absolute game changer for global science. We urgently need that policy push to sequence the remains of our own ancestors. What a perfect concluding thought to leave our viewers with. Aspirants, make sure you use this data critically and thoughtfully in your answers. Thank you so much for joining us for this Rungta IAS Academy Explainer. Keep up the hard work, stay curious, and we will see you in the very next session.